Every single day, millions of us take medicine that we trust. We trust it to make us better, but what happens when that trust is shaken by a hidden flaw? We are about to unravel a scientific mystery that started with just one pill and then spiraled into a massive global investigation, forcing all of us to rethink the safety of our medications.
It's a really unsettling question, isn't it? But it's the exact question that regulators and scientists had to face head-on back in 2018. And the answer would go on to challenge the entire pharmaceutical industry and change how our drugs are made and monitored forever. Okay, let's dive right into this.
So it all kicked off right here, June 2018. A contaminant called NDMA was found in valsartan, which is a super common blood pressure medication. But that discovery? That was just the tip of the iceberg. This timeline really shows how it was the first domino to fall. Pretty soon, more of these nitrosamines were found in other sartan drugs, then in antacids like ranitidine, then in metformin for diabetes, and even in some antibiotics. It became very clear very fast that this wasn't just some isolated incident. It was a systemic problem that had been hiding in plain sight all along.
So let's rewind and start at the very beginning, with the discovery that set off alarm bells all around the world and introduced a brand new, kind of scary word into our conversations about drug safety.
The culprit at the center of this whole story is a class of compounds called N-Nitrosamines. Now, these things aren't new. Scientists have been worried about them for decades. You can find them in food, water, even tobacco. But finding them in pharmaceuticals, where purity is supposed to be everything, well, that was a whole different story.
And here's the basic structure. It looks deceptively simple, right? You've got a nitroso group, that N double bonded to an O, and it's attached to an amine. It's that specific chemical hookup that makes these molecules so reactive and, as we're about to find out, potentially so dangerous.
So, to really get why this sparked such a massive global response, we have to understand the science behind the risk. What is it about these tiny, invisible impurities that has health authorities so concerned?
Okay, now this is where it gets really interesting. This shows what happens after a nitrosamine gets inside your body. Your liver enzymes basically go to work on it, and in that process, they create this really unstable, highly reactive little particle. And that particle is the real villain here. It can directly attack and alter your DNA, causing mutations. This right here is the mechanism that links nitrosamines to an increased risk of cancer.
Because of that DNA-damaging potential, international guidelines have put nitrosamines into this very serious-sounding special category called the cohort of concern. And this is a huge deal. What it means is that the usual safety rules for impurities, they just go right out the window. These compounds are considered so potent that they pose a significant risk even at levels way, way below the standard safety threshold.
And get this, not all nitrosamines are created equal. Just look at this table. It shows a massive difference in potency. On one end, you've got something like NDEA, which is highly potent. The safe daily limit is just 26.5 nanograms. We were talking billionths of a gram. It's an unbelievably tiny amount. Then on the other end, you have the less potent NDPH, with a limit of 78,000 nanograms. This huge range is exactly why each nitrosamine has to get its own specific risk assessment.
Okay, so the big question, how did these contaminants even get there? The investigation turned into this incredibly complex piece of detective work, tracing the journey of a pill all the way from raw chemicals to the finished product you hold in your hand.
The first major clue led investigators right back to the manufacturing process for those sartan drugs. See, to make the process more efficient, a manufacturer changed the chemical recipe. But this new process used different solvents and reagents that, under just the right conditions, accidentally reacted to create NDMA as an unintentional and totally undetected byproduct. It was a perfect chemical storm. But the contamination wasn't just coming from inside the pill. In a shocking twist, investigators found another source: the packaging itself. You know the foil on some blister packs? Well, it can contain a layer with something called nitrocellulose. And during the heat sealing process, that nitrocellulose could actually break down, releasing chemicals that would then migrate from the foil into the pill. Wild, right?
But it wasn't always just one single culprit. In a lot of cases, like with the antibiotic rifampin, it was more of a combination of factors. You could have a tiny impurity in one of the starting materials that then reacts with other chemicals. Or the drug itself might just degrade a little bit over time during storage, creating the nitrosamines long after the pill was even made.
And this might be the most surprising part. Even the inactive ingredients, the fillers and binders known as excipients, could be a source of the problem. Everyone thought these were totally harmless. but tests showed they can contain trace amounts of nitrites, which is one of the key building blocks for a nitrosamine to form. So the risk, it turns out, was sometimes baked right into the very recipe of the pill itself.
So, faced with this complex, multi-layered problem, regulatory agencies like the FDA in the US and the EMA in Europe knew they had to act, and act decisively. They coordinated on a really clear, logical plan to get control of the situation.
They rolled out this standardized three-step approach for all drug manufacturers. Step 1, risk assessment. You have to review your entire process to find any potential risks. Step 2, confirmatory testing. You need to run highly sensitive tests to confirm if nitrosamines are actually present. And step 3, mitigation. If you find them, you must make changes to eliminate the source of the contamination. Period.
Now, while the overall game plan was the same, different agencies took slightly different approaches. Europe's EMA, for instance, has a testing strategy that depends on the level of contamination they find. The FDA, on the other hand, is a bit more stringent, recommending testing even when the levels are very low. These little differences just show there are different philosophies for how to manage risk while also trying to prevent major drug shortages.
And this is a challenge that just keeps evolving. As authorities look deeper, they started finding new types of contaminants. Of the nearly 200 different nitrosamines recently reported by European authorities, look at that number. 176 of them belong to a newer class called NDSRIs. These are nitrosamines that are structurally related to the drug molecule itself, which represents a brand new, really complex frontier in this whole challenge.
All right, so for our final section, let's look at the solutions. Because, discovering the problem was one thing, but the real work is in engineering ways to prevent it from ever happening again.
The industry is now deploying a whole toolbox of mitigation strategies. They're changing chemical manufacturing routes, they're sourcing pure, low-nitrite fillers, they're redesigning packaging to be totally nitrocellulose-free, and they're even adding antioxidant scavengers into the formulas to gobble up any precursors before they can react.
To deal with all the newly discovered nitrosamines, scientists developed a really clever predictive model. Based on a nitrosamine's chemical structure, they can place it into one of 5 potency categories. This lets them assign a temporary safety limit, like the ones you see here, even before long-term animal studies are finished. It's a fast and very conservative way to protect patients.
The ultimate goal here is to get ahead of the problem. So companies are now using predictive software and collaborating on shared databases to track ingredient purity. The idea is to run risk assessments in a computer simulation, purging potential impurities way back in the design phase of a drug, long before it even gets close to being manufactured.
This entire saga, from valsartan all the way to today, has taught us that ensuring drug safety is a process of constant vigilance. It leaves us with maybe the most important question of all. How do we build systems that are smart enough to find the next hidden flaw before it becomes a crisis?
The search for that answer is happening right now, driven by the scientists and regulators working behind the scenes. It's a powerful reminder of the invisible science that works every single day to protect our health and maintain our trust in the medicine we take. Thanks for watching.